Okay, well, welcome everybody. Thanks for joining us. I think this is the second of our webinars in the webinar series from the Water Watch Collective. So I'm going to talk today about using riverflies to monitor water quality. And then it's a bit of a double header. You're lucky enough to have Tony here as well, and he's going to be telling us about some of the riverfly in action that he's been doing with local schools here in Gloucestershire. So I'll kick off with just saying who I am. So yeah, I'm Hannah Robson. I'm based at the Wildfowl and Wetlands Trust at Slimbridge in Gloucestershire. I work in the research department here. I'm the head of wetland bioscience, so I head up a wonderful team of people working on all kinds of wetland ecology on WWT's projects around the UK and internationally as well. And I kind of wear another hat as well, and that is my riverfly hat. So I'm kind of wearing both hats today. But yeah, so all sort of areas of wetland ecology is my bag. And so I guess I just wanted to sort of kick off with a few reflections about where we are and what's happening in the world of water at the moment. And I guess the sad thing is there's some really shocking stats out there at the moment. We're getting more and more used to seeing headlines like these, and it's really, depressing state of affairs that's the way things are at the moment. And it's not just headlines, it is also being backed up by the facts as well. So we've got things here like the Rivers Trust State of Rivers report from 2024, showing that not a single stretch of river in England was in overall good health. So that's that left-hand donut there you see. And when we talk about overall health, we're talking about not just the taxa and the ecological component of it. We're also talking about the geomorphological structure of the river, the habitats present, and also the water chemistry and the hydrology as well. So there's no river in the whole of England that's scoring good for all of those criteria, which is a terrible place to be. If we zoom in on ecology in particular, then the picture doesn't look much better. We've got 15% of rivers that, just on that, when we're just considering ecology, that they're scoring a good. But we've got a vast number of rivers that are not in a good state at the moment. So hang on, just move these people out of the way. There we go. So why is that? Well, there's lots of reasons for failure, and some of them, again, from this Rivers Trust report, you can see here. Some of the sectors that are being attributed for those reasons for failure, the reasons for not having good ecological or other status, and they're mainly coming from things like agriculture and land management. It's a big chunk coming from the water industry, which is perhaps the one we hear the most about. And then that third one, there's still quite a lot that's unknown and that's still under investigation. So those three, top main sort of offenders there really sort of are the reason for some of our rivers not doing very well at the moment. And what they're doing, what's causing this sort of reason for not achieving good ecological status is this. So this graph now is showing the reasons why they're not achieving good. And the main reasons, the dominant reasons, are sewage discharge, poor nutrient management, and poor livestock management. And all of those things together, they really just mean excess nutrients going into the rivers and causing all kinds of problems. And of course, I guess perhaps some people might think, well, nutrients is a good thing. You need nutrients to live. Isn't that a good thing if rivers are getting nutrients? Well, I guess the thing to remember is that too much of any one thing is going to cause a problem, and that's what's happening with our rivers and our wetlands in the UK. So I've just put this up here just to sort of illustrate the difference, what happens when we get these excess nutrients going into waterways. So the excess nutrients is going in, and it's feeding all of those algal blooms that are forming on the water. And the problem with those is that then they're blocking out sunlight. So then the sunlight isn't penetrating the water. The plants and macrophytes that are growing underwater, they can no longer photosynthesize. They're not getting enough light, and so then they die away, and they're not producing oxygen in the water, and they themselves die. And then, of course, they need to be broken down by something. So then a lot of bacteria comes in to break down the dead and decaying matter. And that's just producing yet more carbon dioxide and less oxygen. And then ultimately that is then going to impact on all of the larger organisms, the invertebrates, the fish, everything else living in the water. So it's really important. It might not seem it straight away, but it's really this nutrient issue that seems to be the biggest problem for us in our catchments and rivers around the UK. So what can we do about it? How can we monitor what's going on with our rivers? Well, we can obviously do that in different ways. We can monitor particular nutrients such as phosphates or nitrates by taking samples, taking an actual sample of the water, taking it back to the lab and doing some measurements, or we can do measurements in the field as well. But there are quite a lot of challenges around that, one of which being cost. It's obviously quite costly to send samples away to the lab, and even the quite basic field kits, the costs can start to rack up. But another issue with water sampling is the issue of timing, that you could have a pollution event in a river, say, that had sort of a pollution event happens, it passes down through the river, and you come along the next day with your nutrient probe or whatever, and you find, oh, well, it looks fine now. It's, it's the sort of nutrients has been washed off down the river, and you're not really sure. It makes it difficult to know where and when the pollution issue might have occurred. So another approach that we can use to, in a lot of ways, complement those water chemistry measurements is to actually look at the organisms living in the river that are sensitive to those changes. And that can be really helpful for a whole number of reasons, which I think I've got on the next slide. But it's really the ones that we are going to focus on are the ones there are several taxa that are very impacted by or sensitive to changes in water chemistry. And by monitoring those, we can get an indirect understanding of what's happening with the water chemistry as well. So why do we use invertebrates? There's lots of advantages to using invertebrates to monitor water quality, and that's why they're such an important component of the Environment Agency's and other statutory agencies' routine monitoring. The good thing about invertebrates is that they're present in almost all freshwater habitats. They can be supported to some degree or another, even in quite hostile habitats. But you just tend to end up with a much smaller list of taxa that are adapted to cope with those extreme conditions. As I mentioned, there's lots of different species, and really importantly, we can identify them, which is something that's quite challenging for other things, for example, diatoms or algae. Yes, there's a lot of work been done, but they seem to keep discovering new species and new keys and new taxon splits every 5 minutes. So I think the good thing about invertebrates is there's lots of different species, but there's been a lot of work done to identify them. There is more still going on with DNA and different things as well. But each one of those species have a different sensitivity or tolerance to different conditions. So some might be more sensitive to low flow conditions or high levels of nutrients or low oxygen, and those ones that can't cope with those changes in conditions, they're not going to be present when those conditions are present. So they're really useful. We can work out what they are, and we can work out what they're sensitive to. I've also put up on this slide, they don't move around a lot. I guess I'm not saying that they just sit around and don't do anything. Of course they don't. They fly around and do all kinds of things in their busy lives. But I guess what I mean here is they don't migrate really, really large distances. So if you find a species in a certain place, it's a relatively short range from which it can have come, meaning that your samples that you take are going to be spatially and sort of temporally relevant. And I guess the other thing on the temporal side is that many aquatic invertebrates have life cycles that are months and in quite a few cases years. So they spend months and years as their aquatic larval stage and then spend a very short amount of time in their adult terrestrial phase. So again, it's quite useful in that sort of working out that temporal component to what's going on in the water at that time. So I wanted to talk today a little bit about Riverfly, because they're another great reason why I love invertebrates, and I think why lots of people love invertebrates is because they're really accessible and they're engaging for everyone. Well, maybe not everyone, maybe some people don't like them, but so pond dipping for a lot of us was probably quite a common activity carried out by us in our childhoods. And I guess the good news is we now have lots of reasons to do it as adults as well. We can get involved as citizen scientists. We can monitor our rivers, and we can sort of take action to address some of those alarming sort of headlines that we saw at the beginning of the presentation. The scheme I'm going to talk about was developed in conjunction with the statutory agencies like the Environment Agency. So the method itself and the way that the scores have been developed and all those sorts of things, they're all sort of benchmarked and sort of tested very thoroughly against full species level samples taken by the Environment Agency. So they would take a sample and go through the statutory agency process, and at the same time they would take a riverfly sample and go, so they'd be able to sort of benchmark what we were finding. So it's a really robust method, but the nice thing about it is it's really accessible, and it just so the Riverfly Partnership aims to sort of bring together all of those people interested in water quality, riverflies and their habitats. So definitely check them out. So I guess lots of people say, is the Riverfly Partnership, is it an organisation, is it owned right, is it part of a umbrella under something else, how does it work? So I thought it was useful maybe just to sort of lay out the structure of the Riverfly Partnership, just to sort of so people can get their heads around how it all fits together and then where you can get involved. So it has this kind of nested structure with the Riverfly, the National Riverfly Partnership headquarters that are providing ongoing support, technical help and communication. That's quite a small team. I think it's maybe 3 or 4 people. It's a very, very small team. And they're hosted by the Freshwater Biological Association up at Lake Windermere. And they do, as I say, the sort of national level coordination and support and provide the training materials and all that kind of thing. And then at a regional level, we have these hub coordinators, and that's what I am. I'm one of those for the Severn Vale, but there are lots of those around the country, and on the whole sometimes they are people based at a ENGO, but sometimes they're just people, professional or not, who are knowledgeable enough to be coordinating riverfly at a sort of regional scale. And again, so their remit there sort of expand the groups monitoring within their area to be a sort of point of contact for group coordinators, and to provide training and ongoing support and things like that. So that's the sort of hub coordinators. And then below that, there's often, not always, but there's often a sort of group coordinator. So that might be along a particular stretch of river. There's a group of 10 or 15 citizen scientists who are all working, and they kind of have a couple of people who are coordinating them to say, and that often just volunteers themselves that are saying, okay, we'll do this bit this week, you do that one next month, blah blah blah. So it's kind of, there's that level as well. So they're really important. It's really sort of all the way from the bottom up. It's really important. And then off to the side, sort of supporting the hub coordinators and the group coordinators, is the ecology contact from the Environment Agency. And this is a really important role, and we're really lucky in the Severn Vale to have a really proactive ecology contact, Pete Smith, at the Environment Agency. But they're really good. They are a point of contact for the hub coordinators and the group coordinators. They often attend the training sessions with us, so it's really good to have another set of hands in the room to offer some different perspectives. And then they also sort of help us set the trigger level, which I'll come on to in a second, for the different sampling sites that we set up as part of Riverfly. So really important that it's sort of all bedded in with the statutory agencies as well. So I'm not going to spend ages on this, because this is really sort of going into probably a bit too much depth for the training, but I just wanted to say sort of what a trigger level was. So what usually happened and how you sort of set up a site. So a Riverfly Monitoring Initiative hub or group or individual wants to set up a new site. So they need to sort of consider all of these things in that top box. They need to sort of think about health and safety and their volunteers, but also more sort of fundamental things about habitat diversity and the types of conditions present there. So there are certain things that we want to be looking for when we set up a new Riverfly site, and that's something that I can help with, certainly locally. And so then we agree that that's a useful site to be setting up, and we fill in a bit of paperwork. We send that to the ecology contact at the Environment Agency, and then they look at their historic data for that catchment, and they say, right, okay, well, if based on what we found so far in that catchment, what we don't want the score for this site to go below 4 or 5 or 3 or whatever it might be. So they set this kind of level, and that's obviously based on a lot of previous data, and that sort of sets the bar below which we get very worried if things fall below that trigger level. So then we send the site information to Riverfly National, and they set it up on this platform. That's what Cartographer is. It's just a platform that we use to enter the data. And then we can then the volunteers can begin monitoring ideally on a monthly basis. So that's sort of how it all works to get you to a place where you've got a site set up and you're ready to go. The bit that's probably sort of more interesting is what do you actually do when you get out there? So the first thing that we need to do is collect our sample. And again, so all of this to do the Riverfly training and to have a site set up and to get your data registered on the platform, it's really important that you do the formal Riverfly training first, which locally I can provide. And if not, then we can put you in touch with whoever your local coordinator or local trained trainers might be. But so this is what you will learn when you're doing a training session with us. You will learn how to collect your sample and what you need for that. So it's a 3-minute kick sample, and you will realize how long 3 minutes is when you're there kicking away, and you think it's not that long until you're there knee deep in sediment. And there's a kick sample component, and then there's also a stone turning component, and it's sort of looking at stones, picking up some stones that are sort of fist size stones, and washing off any things that are really clinging onto those stones into the net as well. So there's collect this sample, and then as you can see in the equipment on the left there, you put all of your sample into this white tray. And the key difference there, and that is exactly the same method that the Environment Agency will use, that 3-minute kick and those stone turns. They will then take their sample back to the lab and spend a long time looking at things down a microscope and taking things to species level. The bit where we differ is we do all our sorting on the bank side. So we look at our trays on the bank side, and we might see some things like this in there. So these are just some invertebrates that we might see in there. And usually when I flash this slide up at training sessions, usually people, when they first see it, they think, oh my God, there's so many there. And then actually when you run through it, usually people can identify quite a lot of these already. But the ones that we're focusing on for Riverfly, standard Riverfly, are just these ones here. So again, so if you were daunted by all of those different ones, the ones you need to know for Riverfly are these ones. So in the top middle there, we've got a mayfly with the 3 tails. We've got on the bottom left there, we've got caddisfly. We've got a cased caddisfly there on the right-hand side, and the caseless caddis there on the left-hand side. So they're really nice. They produce a silk, which they either use to construct that case that they're making, that one on the right is making, or the one on the left is a more free living kind, and it constructs all kinds of really cool sort of nets and webs in the flowing water and catches detritus as it's flowing past. So they're really cool. There's a couple of different kinds of them that we look out for. And then above those with the 2 tails there, that's a stonefly. So they're very, very intolerant of poor water quality. So if you're getting stoneflies in your sample, you know you've got a site with good water quality there. And then, although it's not a Riverfly, we let it sneak in anyway. It's the freshwater shrimp there as well. So that one on the left-hand side at the top there. So they are the ones that we are looking for when we're doing our bank side sorting. And then when we're doing our bankside sorting, we then categorize, we identify those different taxa, and then we give them an abundance score. We give them a score based on their abundance. So again, these sort of bands of counts have been sort of what's the word, benchmarked against the Environment Agency sort of sample. So those, although you sort of think, oh, there's quite a difference between 100 and 999, actually they've sort of done quite a lot of WYSI stats that I didn't understand in the background to work out that these are appropriate bandings for us to be able to pick up trends and differences. So that's what we tend to use. And then what we do is that helps us calculate a total score for that site. And then we obviously can monitor that, and then there's just a little graph there in the bottom right to show, that red line going across is an example of a trigger level. And then, if we see the blue line, which is the score for the different Riverfly samples taken, if we see that score fall below the trigger level, then that's a point at which we would be very worried and taking action to do something. And again, this is a day's training in which I could go through in much more detail all of the sort of steps that we could take to raise the alarm and get action taken for that. So there are actually 3 different kinds of Riverfly survey at the moment. It's grown, it's doing really well. What we've got at the moment is standard Riverfly monitoring, which is 8 taxa. So there's the 4 different kinds of mayfly there in the top right hand side of the slide. So they've all got 3 tails. They've all got 6 legs. They've got varying amounts of gills of different types. And so in standard Riverfly, we will help you identify those 4 different kinds of mayfly. We'll help you identify the cased caddisfly there, the caseless caddis, the freshwater shrimp, and the stonefly. And they're the 8 taxa that will help us to detect severe changes in water quality. They're particularly sensitive to nutrient changes, these ones. So they're the ones that will help us work that out. And then the other thing that sort of spun off from that initial basic Riverfly was urban Riverfly. So I think what we were finding was in some of the very degraded urban catchments, people weren't finding a lot of those 8 taxa from the standard Riverfly, and it was getting a bit sad for them, really. So we started to think about, we want people to be engaged, we want people to be champions for their rivers. So let's sort of broaden out the scope a bit, and also let's sort of include some species that maybe score negatively as well as positively. So we've got some things on here like the black fly larvae and the freshwater hoglouse and the worms and things, and they are very tolerant of poorer conditions. So this urban Riverfly is a kind of add-on from basic Riverfly and includes some extra taxa and maybe more suitable for some places where we're not getting the full range of the 8 there. And then, of course, there were people who were so good at basic Riverfly that they were getting a bit bored and they needed more to do. So there's been quite a lot of work over the last probably more than that now, several years, to bump up the Riverfly list and expand it into an extended version. So this extended version includes 33 taxa, including the 8 from the original standard Riverfly. So you've not wasted your time by learning those 8, because those 8 are in urban and in this one. And the nice thing about the extended Riverfly is that it includes not just taxa that are sensitive to water quality changes, as in sort of organic pollution and things, but it also has picks out taxa that are sensitive to things like siltation, which is a big issue for a lot of our rivers, and also extreme flows. So some of the taxa that can't really cope with very low or high flows. So it can give you much more information. It's a more in-depth training course, and I can train people in it. But I would recommend that people get very comfortable with standard Riverfly first, and then we move on to extended Riverfly. So it's a kind of carry on from standard Riverfly. I was just going to touch on a few examples of sort of Riverfly in practice, really, and just to sort of demonstrate how it can be used to address some of the issues being faced by our rivers. So they're from all around the country. This first example is from the River Itchen. The site's been identified as really important for salmon spawning, and the water quality was being regularly monitored by Riverfly volunteers. And then one month, a volunteer turned up on site and noticed that there was huge amounts of very fine sediment entering the river, which obviously could have had very severe effects on the spawning, the clean sort of gravel spawning areas of the salmon. So the volunteer was able to contact their ecology contact at the Environment Agency. They would be able to provide the previous data from that site to show that it hadn't been like that for previously, and to show that there'd been a significant change. So I guess this one's just a sort of an example of the value of having people out on rivers and having that regular presence and that regular sort of eye on the river. Because something like that could have gone unresolved. It turned out it was there was a landowner upstream who'd basically it was a land management issue slightly upstream, but that potentially would have gone on for quite a long time, and potentially obviously affected generations of salmon before somebody properly noticed it. So it's really sort of just showing the value of having people out and about advocating for rivers, and maybe that's one of the ways in which we can support making change happen. The next example is a bit more of a recent example. I think this is from January this year. I think it was this year. Yeah. So this is up in County Durham, and it was a sampling scheme that was being run by the Weir Rivers Trust, and the site was being regularly monitored. It had a trigger level of 4, so they were monitoring away, and it was all above 4, and it was doing okay. And then they did one survey and they got a score of 3, so they only had 30 olives and 9 shrimp in there. So what we do when we first get a trigger level breach, as we call it, we go out and take a repeat survey just to make sure that what we're finding is a true reflection of what's happening, and we didn't just sort of have a hole in our net or have some issue that we perhaps didn't clock the first time. And the second survey had very similar results, a few more shrimp, but it just about managed to scrape a trigger level of 4, a score of 4. So there were some conversations with the Environment Agency and the ecology contact about ongoing concerns about this river, and again the volunteers being out on the river, they walked a little way upstream and got a very strong smell of detergent. And this one was actually there was a culvert. It was coming out of a culvert just upstream with a really strong smell out of it, and quite a lot of foam, which is what you can see there in that bottom picture. So the Environment Agency got in touch with Northumbrian Water, and it was actually sourced back to a single household who'd recently had some renovations done, and I think they just the builders had sort of misconnected the downpipes from and their washing machine had gone into the rainwater drains and things. So it was, again, really quick action because we had people out there monitoring month in, month out. And it was sort of because we had that evidence and that data, the Environment Agency were able to sort of take action really quickly. So now that site's being continues to be monitored, and hopefully we'll see an uptick in those scores as we go through this year. And then this final one, I just wanted to sort of use as an example just to show that riverflies don't have to just be used to monitor bad things that are going on in our rivers. They can also be used to monitor successes. So here on the left, we've got a before photo showing a site up in Warwickshire that was quite homogeneous. It was quite a sort of incised channel, quite a straight, uniform flow, very sort of homogeneous habitat types and flow types going on there, and quite a bit of silt and algae. And Warwickshire Wildlife Trust, in conjunction with partners, they did some work on this section of river. I think it ran behind the back of a hotel there, and they were taking steps to improve the diversity of flows, which also has the knock-on effect of improving, sort of diversifying the habitat, and which is what you can see there on the right-hand side. So they used Riverfly in this scenario before they'd done anything. And the results there showed a very low density and low abundance of taxa. And then they're currently monitoring on the right-hand side there. I mean, you can already see the water's quite a bit clearer, but they'll hopefully be seeing that in their Riverfly scores as well. So I think it's sort of nice just to sort of touch on it. We don't have to have it all doom and gloom. We can also use it for really positive things as well, sort of demonstrate successes as well. So hopefully I've excited you about the potential of Riverfly, and you all want to get involved straight away. So the first thing you need to do, if you're keen to get involved, is attend a training session. So locally in the Severn Vale, these are going to be advertised on the Waterwatch Collective's website, or contact me direct. But in other parts of the country, check out your local Rivers Trust, Wildlife Trust, other NGOs, or if you can't find anybody locally, then definitely get in touch with the National Riverfly Partnership, because they will be able to point you in the right direction to get some training and get set up. And then once you've got yourself trained, you can either sort of join an existing team or project or organisation. That's often the case that ENGOs have already got work monitoring sites already set up, and you can join them, or you can work with your hub or group coordinator, not your grub coordinator, to set up a new site. If you've got something in particular, maybe there's something happening in your local stream and you're really worried about it, so the thing then is to have a chat with them and get that new site registered on the with the Riverfly National. Yeah, so again, there's this sort of a Severn Vale examples there, some of the organisations that are involved in bits of Riverfly and are supporting via the Waterwatch Collective. And then once you've had the training and your site is set up, then you can start monitoring and uploading your data. So here's some very happy Riverfly volunteers. And there's just an example there of the sort of form. And then in fact, I've got a better map on the next slide, actually. Then you can start uploading your data. So here is the platform cartographer that shows some of the sampling points that are set up locally around here. I know perhaps some of you aren't all local, but there's quite a few. All the ones in purple here are ones that are, I think they're the ones that have lapsed. I think, Tony, you do some of those green ones, don't you? So they're yeah. So I think those so that I was trying to remember what my color coding was there, but I think the purple ones are ones that were historically set up, but the volunteer are no longer being monitored. So anybody who would like to pick up any of those ones, get in touch. So yeah, and then through the cartographer platform, you can explore the data for your area. You can look at what the scores have been historically. You can look at trends and all kinds of things like that. So yeah, it's a really useful platform as well, but can provide you with either some training on that at the Riverfly training day or the Riverfly Partnership National. They do some one-off training sessions on that as well. So I can set people up with that as well if that's helpful. I guess if I put you off Riverfly in the last half an hour and it doesn't sound up your street, then I guess the other thing to say is that there are other things that you can do. We've sort of joined forces with these other citizen science initiatives, Morph and Freshwater Watch and Outfall Safari. And they are looking at sort of different components of river health. So Morph is sort of looking at the geomorphological structure of rivers and looking at different types of flow. It's probably slightly, I want to say slightly harder, but that's maybe doing it a disservice. I sometimes do a double header training session with Riverfly and Morph, and I think people sometimes find the Morph a little bit drier, but it's very important, really useful stuff to do. It's sort of much more observational, and getting to know different habitat structures and vegetation types and things like that. Freshwater Watch involves taking some in-field water quality measurements using little test kits. So they're sort of little colour changing kits for nitrates and phosphates that you can take in the field. And I think they've got kind of bespoke Secchi disc tube and various observational data associated with that as well. And then Outfall Safari was developed by ZSL, and that's looking specifically at outfalls and results of misconnections. So started over in the Thames catchment, but I think is now spreading out all around the country. So yeah, if you're not up for Riverfly, then there are other schemes out there that might float your boat. And I guess finally, I just wanted to say for Gloucestershire folk and Severn Vale folk, definitely worth being aware of the sort of fairly newly formed Water Watch Collective, which is a group, a sort of coalition of ENGOs and statutory agencies and water companies all coming together to try and sort of consolidate knowledge on citizen science and support a sort of regional scale initiative. It feels, I guess our sort of motivation for this was, it seems like there's a lot of national scale schemes like the Riverfly, which is national, and then there's quite local sort of single catchment based schemes and projects that are very sort of focused on a single stream or stretch of river perhaps, and really sometimes it can be quite useful to think of things at that regional scale and to sort of zoom out slightly. So we're trying to fill that gap a bit, and Charlotte's doing a cracking job bringing all that sort of information from all those different sources that are out there. I know you can search the web for ages and find all kinds of guides to citizen science and all those sorts of things, and Charlotte's bringing them all into one place for you. So that's what's there on those websites there, and there's also a data portal for Gloucestershire which brings together all of the data from the Riverfly, from Morph, from Outfall Safari, from all the different and the statutory agency data as well, brings it all into one place so that you can look at a regional scale and see where the gaps are, where, see what's going on in your local patch. So definitely worth having a look at that as well. I think that was all I was going to say. So I think now I will hand over. Shall I hand over to Tony, and then we can have a bit of a breakout chat at the end? Does that sound all right?
Yeah, I think that'll be good. Do it all at once then, and I'm conscious of people's time also.
Sorry, I've managed to talk on.
Okay.
No, it's very informative.
Thank you. Just a little bit of background. I work with the community engagement team for the canal restoration project in Stroud, in case you're not familiar with that. And part of that means that I spend a lot of time working with schools, but also with lots of other organisations. Schools have approached me wanting sort of, to know how anything that's going on. And whenever I've mentioned the Riverfly monitoring, they are absolutely fascinated at the idea and are keen to get students involved in some way. And when we've given that opportunity, it's so engaging. And all I want to do is to show you 2 or 3 examples of what we've done. So I'm just going to share this and hope it works. Can you see the screen?
Yes.
Yes.
Okay. Right. So the first example was we had a secondary school approach us, and they wanted to do something slightly outside of their curriculum. They wanted a project, a STEM project, and the award is called a Crest Award. And they just needed to do an extended period of research and work as a team and do a presentation at the end of it. And they were all really excited about the Riverfly project. And what we did to give them an extra bit of challenge, we said, yes, but at the end of it, you are going to do a presentation to an audience that we are going to select of adults, and that's going to be in the council chambers. And that's where they are here. They were really scared when we mentioned it, but by the time they got to it, they were kind of, well, you can see, fully confident. So what did that involve? Sorry, that's them doing their presentation to a really tough audience, right? Who asked some very challenging questions. So they asked me to come to the school for one visit, and that was really so that they had some time to have some questions. So I didn't have a presentation or anything. It might look like I have, but they just literally had questions, and I wanted to show them that they would have access to some data. And that's what we've got on the screen. And that's all the Riverfly data nationally, but it allowed them to have a question, and they wanted to come out and measure something themselves and then compare it with something else. And it was a nice, simple idea. And I just had to take some data out of that site and just send it to them, and they got on with it. This is one of the students on site when they came down. So as well as me going to the school once, they came and they did 2 site visits where it was about a 20-minute walk from the school. That was a major thing for the school to give them time to do that. But because they had a very supportive leadership at the school, then we could arrange a date for that to happen. And they're very limited in time. So what we did is they came up for one visit where they did the identification. So we had already taken a sample, and they came and had half an hour getting really excited about what they could find. And then they came a second time where we took them into the river and let them have a go at sampling themselves. And it felt better to do it that way round. It sounds wrong, but the sudden discovering of these creatures and being successful at identifying them is the bit that really hooks them. The bit they remembered and talked about most was being able to go into the river and do the sampling. But I think if it was the other way around, it might not make as much sense to them. So that was one school with their own little project. And then because of that one, we had another school approach us when they heard about it, and they asked if they could be involved. And they're so keen that we ended up training one of the teachers there so that they could join the volunteers and monitor it themselves. And this is that school on a different river. They're not quite sure what to do. So we tried some things out, and then we got ideas for next year. Again, they've found it easier to get the sixth formers involved, and it's not because of the level that they're working at. It's simply because they're smaller classes, and they can experiment a little bit. But this is the great thing. So we brought a sixth form group down to do some sampling of the river and to go back to the class. This is one of the things that excites them most, is when they see this, what might look like a log or a stone, and it suddenly comes to life when they put it into one of these trays, and it's an image that stays with them for ages. This is how they're working in class. So they went through this whole thing, and they were told that they would then teach a year 10 class. And I haven't got photos of that, I'm afraid, but we went back with the sixth form groups, who are really motivated because they're doing something even more challenging as a result of it. And the teacher from the school couldn't believe how successful that worked. And she's ended up, just from that one experience of the sixth form. So we took a sample out of the river to get into the school because it wasn't that far. And then the year 10s were able to do the identification part. They've all now signed up to do biology A level, even though it's not for 2 years. Because they just think that this is a world that they want to be involved in, and teacher said it normally takes a lot of work to get people to commit to doing biology, and she's just done this standing back almost and allowing the sixth forms to just engage those students based on this project. And it's also, it always comes with this big picture. So not only are they doing something that they're fascinated by, but it has such importance in the world, and they're being reminded all the time in the news when that you don't have one day going without some water quality incident being talked about. It's not just the older students though. So here is our a year 4 class. So they're actually doing geography, and they asked us if they could, if we could help them with some field work. So we said yes, we've got a river near us, and what is it that you want to do? And we got a bit of information from the curriculum, which was all about, they need to know about river features. And we said, well, why don't we can do 3 things with you? Okay, we've got plenty of river features. Why don't we look at a natural feature, a man-made feature, and then life in the river? And that sounded perfect. And it was so good that they were prepared to walk to the site, which is about a 2-hour walk for this school. Yeah, I know it amazes me, but they said no, that's fine. It's a day out. The person with the white cap on there, so he's a Riverfly Monitor volunteer. Anyone who is a volunteer for this is an expert, especially in the eyes of these children. And so long as there's a little bit of structure provided, then they are just doing what they do, which is sampling from the river, identifying things, and being enthusiastic, which I've never met anyone involved in this who isn't. And that's what we have. And here, sorry, this is just the different elements of it. We didn't take the little ones into the river for this. That's another activity. They can, but it's just that was too much for one. So we just let them do the identifying. But I think just looking at them, I think you can see how engaging and fascinated they are. It was a bad day for us. We were quite concerned, right, because we were at a trigger point on this day, and we had to go back there just to make sure. We couldn't rely on what they were finding, and unfortunately, it's been like that for a while, and we haven't quite. Don't quite know what's going on, and this is the group just on another little section of the river. But that's you. So that's not a kind of the younger children. We've had requests from all kinds of things. We've had a beavers group ask us, based on what they've heard, if we could come in and talk to them and show them about it, who are even younger than these children. And again, the great feedback that we had from that one was about them, what their parents thought. So they go home and they talk about this thing that they were kind of, and all, they've just been identifying invertebrates. And we've had, I haven't got quite the right picture, but I've got the students there. So that's a couple of students from a special needs college who came to the same site to do the Riverfly monitoring. And again, it's so, as Hannah said before, it's such an accessible thing that anyone can be successful in identifying the different invertebrates, and we can make it more or less accessible depending on the needs. So I'm going to stop there because it's just to give you a flavour of what we're doing, and what we intend to do now is a little bit more experimenting with schools who are flexible enough to be able to do that and have their own close enough to a site. And I think that over the next year or so, we'll probably be able to come up with some sort of really kind of good guidelines or something about how schools can get involved with this in so many different ways. just going back to those secondary schools, the first one that were involved in quite a major project, they only came into the river for one session and only did sampling for one session, but that's the bit that stood out most for them, even though they probably spent about 30 hours doing a research project. But without that kind of hook and that experience, then the whole thing just, you wouldn't have got that age group, I think, to invest in that kind of research. Okay, I'll stop there.